Now in US it is summer, right?
Yes, and then in Texas, it never gets cold. So it's always summer here. It's
Oh,
very,
yes, I believe, yes, yes. Just like in Florida, Florida also.
yeah, it's just
Mhm.
always very warm. So when I came to the US, UK was, I would say, like pretty good in terms of it would rain very often. But when I came to the
Mhm.
US, I was expecting a little bit of cold weather, but it was very hot in Texas all throughout. Maybe like in winter, we get 1 day of snow, and that is it.
Oh, yes.
Yeah.
Shall we start now?
Yes, we can start.
Hello. So first of all,
Sir, I
hello.
think you're on mute. Yes.
Yes, yes. Uh, Very good evening to one and all. I welcome Dr. A.M. Deshmukh sir, President, Microbiology Society India. I also welcome the today's resource person, Abantika Ghosh, uh, who deliver his talk on the invisible conversation, how microbes shape our immune system. So first, it is our duty to introduce the resource person. So she done her BSc in microbiology from University of Kolkata. She done her post graduation in uh,
I'm sorry, I think you're on mute.
Yeah, we can't hear him.
hello. Am I audible? Hello.
Yes. Yes, you're audible now.
So she done her post graduation from Oxford University, and she pursuing her PhD in biology, neuroimmunology from Texas A&M University, United States. She also have various publication to her credits. She has a broad research experience, and she is working on integrating acute and chronic inflammation after spinal cord injury as her PhD topic. So I request Avantika Ghosh to show his expertise in that subject. Over to you, ma'am.
Thank you. Thank you so much, sir. So yeah, I'll just like again like thank Dr. Vasim Patil for reaching out and all of you for attending today's talk. And I'm very grateful that the Microbiology Society for India is starting up these initiatives, which I think is very, very useful. So, today I want to talk about what I call the invisible conversation, which is the constant exchange between the microbes that live in and on us and the immune system that has to decide every moment what to tolerate and what to fight. So, for the interest of time, if you have questions, if you want to put it in the chat box, I can keep looking at it and I'll try answering it while we are going ahead. If there is anything that I don't know, I am definitely going to get back. So, before delving more into the topic, I wanted to briefly introduce a few things about me. So, I did my bachelor's in microbiology, like so I mentioned, from Scottish Church College in University of Calcutta. And then I went ahead and did my master's in integrated immunology from the University of Oxford. And following that, I also was working at Karkinos Healthcare, which is in Mumbai, after which I decided to do a PhD in neuroimmunology at Texas A&M University. I'm currently doing it. So my research topic is very different from what I'm going to talk about. So I work on looking at acute inflammation after spinal cord injury and how that relates to tissue damage and wound healing. Happy to even like answer questions regarding that. If we have students here in the audience, if you have questions about my journey or anything specific to that, I would be very happy to answer that as well. Now, the reason I chose this topic is one, because I wanted it to be microbiology focused, and because I have a background in microbiology and I really like immunology. So I wanted to integrate both of them together. So let's start by challenging the simplest idea about microbes. Are all microbes bad? So clearly we know that it's not the case, because microbial behavior is very contextual. An organism can be harmless in one body side but harmful in another. So maybe it can be tolerated in a healthy person, but it can become dangerous if the immune system is suppressed, which happens in case of opportunistic pathogens, which usually are fine if conditions are healthy. But once there's an opportunity, then the microbe can surface as a bad microbe. And then we obviously have pathogens, which are always mostly on the bad side of things. So I want us to remember three ideas from here, which is location, context, and the host response. I am going to throw an open question out there, that if can anyone think of an organism that is usually harmless but can cause disease when the immunity is compromised?
एसबी वाला हर चीज में देता है 10%। क्या 10% ऑफ देता है हर चीज पे एसबी वाला। कौन आदमी? केमिस्ट। केमिस्ट
Um,
हाँ मेरे को जानता है वो क्या है। नहीं ऐसे नहीं देता ये। नहीं मेरे को देता
Others can keep their mic mute, please.
है। एक दिन तेरे को इंट्रोड्यूस कराऊंगा ना मैं। इंट्रोड्यूस नहीं करना भाई, सैनिटाइजर बाहर से मंगाऊं कि ऑनलाइन मंगाऊं?
okay. So one of the examples is going to be Candida. Now, before I begin talking about like more complex stuff that I wanted to discuss, I just want to like emphasize on a few other terminologies. So the microbiota is the community of microorganisms living at a particular body site. Um, I can mute. It's fine.
Hello,
Okay.
you can do that, right?
Yeah, I'm muting.
Okay.
Yeah. And the term microbiome is used more broadly to include those organisms and their collective genes. So we have microbial communities on the skin, in the mouth, airways, and also the gut. And the gut is a place where the immune system really faces an extraordinary challenge. And we'll talk about it in details, because it must remain ready to respond to pathogens while also trying to avoid attack on food that we eat, beneficial organisms in the gut, and, and the barrier tissue itself. So the final thing from here is that anything that disrupts that homeostasis or that balance can tip off that system to cause microbial dysbiosis. So moving on to a very brief overview, the relationship between humans and microbiome is that it's more often mutualistic. The host provides habitat and nutrients, and in return, the microbes would expand what we can metabolize. It can help resist colonization by pathogens or the bad microbes and generate molecules that can influence this barrier and immune function. So this is why I prefer the word conversation rather than simply saying the microbiome affects the immune system, because the immune system also shapes how the microbiome exists. Now, I want to show a video. I'm going to see if it plays, but let me see. Okay. So I'm just going to play this video. So when a microbe enters your body, certain cells in your body fend off these microscopic attackers. So early responders like macrophages, they would attack these invaders, and they also give off certain chemical signals that you're seeing, which are called cytokines, that cause reinforcements like neutrophils and natural killer cells, which are other immune cells of the body. Now, these cells consist of the innate immunity. These are non-specific and does everything to contain the infection. But what happens if this infection cannot be contained by the innate immunity? So when it slips through it, you would need a more targeted defense, which is where the adaptive immune system comes into play. These dendritic cells that you're seeing, these are like intelligence specialists that collects pieces of the invaders and then they take them to the nearest lymph node of the body. So inside the lymph node, this dendritic cell is going to present the pieces of the invader to helper T cells. T cells are part of the adaptive immune system. Now, if you see how the T cells bend, the T cells will go ahead and activate B cells, training them to form what we very commonly know as antibodies. And these antibodies target the invaders very specifically. Now, the immune system mounts its ultimate defense where these B cells secretes millions of antibodies, each neutralizing an enemy. So the immune system will keep on deploying these until infection is completely wiped out. So to also simplify it, I would again show you show you my slide. Let's see if I can go back there. Just a second. Okay. So for students who are here, I would want you to imagine the innate and adaptive immune system this way. So imagine you're walking down a stony path and the sole of your shoes tear off. So the first thing you do is you don't want to look for your favorite perfectly fitted pair of shoes. What you need at that time is immediate protection. So you would want to find probably a plastic or like a sandal from somewhere to just wear and protect your shoes. That is your innate immunity. It is fast, it is generic, it is good enough. It will form that layer of response, but it is not specific. Later, what you can do is you can go out using those chappals and go to your favorite store and buy a shoe of your choice that is tailored for your maybe like shoe size, your whatever you like wearing or whatever brand you prefer. That's like what adaptive immunity is. It is much more slower, but it is precisely tailored to this particular pathogen that is entering your body. I wouldn't go into too much details on this, but I would want to emphasize that there is a crosstalk between the innate immune system and microbiota, and these are some examples of how that happens. Now, innate immunity, it includes the epithelial barrier, it includes mucus, antimicrobial peptides, and receptors that can recognize different microbes. So, in the gut, there are immune receptors that constantly receive these low level signals from common cell microbes. Now, these signals help maintain barrier integrity and it also regulates antimicrobial defenses. So, for example, one of them here, if you see, is called the Paneth cells. So, Paneth cells are specialized epithelial cells at the base of the small intestine crypts and they release antimicrobial peptides, which will prevent microbial overgrowth. The next one you see is called pattern recognition receptors. So these are immune system sensors that usually identify pathogen associated molecular patterns, such as toll-like receptors or nod-like receptors. And then they sense these microbial molecules and act accordingly. So overall, what I wanted to emphasize is that the immune system's job is to maintain that boundary, to keep microbes where they belong, to tolerate some appropriate signals, and then react quickly if a pathogen starts invading. A little bit on the crosstalk between adaptive immune system and the microbiota is that, especially in the gut, one major player is the B cell. So B cells produce what is called secretory immunoglobulin A, which is an antibody that traps microbes at the mucosal surfaces. And then we also have T cells, which are equally important. We have regulatory T cells, one of the key players in the gut. That helps tolerate harmless microbial and food derived signals, because imagine that your body is reacting to everything that is foreign. That would be pretty bad, because if you're eating something, if your body is like, no, that is a foreign object, then your body will start reacting to it, and then that is not something that you want. So Treg cells, they help the immune system tolerate these signals, which are everyday signals. Now, Th17, another type of helper T cells, their responses usually are protective, Yet if there is excessive Th17 activity, that can contribute to inflammatory diseases. Now, microbes therefore are going to influence the development and function of these cells, while the cells themselves will shape the microbial community that remains in the gut. And because this immune education begins very early, the first microbes we encounter can be very important in shaping how the immune system learns tolerance. Which is why I want to ask the room that do you think babies are born with microbes or do you think they acquire most microbes after birth? I'll just pause here for a few seconds if any one of you want to answer. There's no right or wrong answer to this question. Anyone? Let me see the chat. Both?
Second one.
Yes, sir. Sorry, your voice was cutting.
Ma'am, I think they get both from birth and artificially are also introduced into them.
Okay. Yeah, I think you're going the right way, and I would say the consensus on that, I would talk about that, is when microbial DNA, it has been reported in the placenta and amniotic fluid, but it is very difficult to conclude anything because these are low biomass samples and they can be easily contaminated during collection. So at present, the strongest evidence that we have is that there is no stable microbiome in the healthy fetal tissues, but there have been traces of microbiome that has been found. So maternal immune signals and microbial metabolites can still influence fetal development, but the consensus is that the major wave of the stable microbial colonization, it happens during and after birth. So during the process of delivery and after birth. So very quickly looking at what is happening here is how a baby is born can actually shape its very first microbial exposure. So, vaginally delivered infants, they usually pick up lactobacillus dominated communities on their skin, gut, and mouth, whereas C section babies, they get colonized instead by skin and environmental microbes like Staphylococcus. Now, these early differences can persist for a time, but they do change substantially as babies start to feed. There is antibiotic exposure, there is household environment exposure, and then they also start feeding on solid food. So, I also want to emphasize, although C-section babies have more number of, I would say, microbes like Staphylococcus, C-section delivery is often medically necessary, like we know. So we cannot really conclude one is better than the other. And all the observed associations with later disease that we see, that does not prove that delivery mode itself is the cause, but it is a plausible explanation. Now, coming to the postnatal factors. So, antibiotics are most commonly prescribed drugs for children, but early exposure to antibiotics can definitely disrupt the developing neonatal microbiome. Now, it's been linked to higher rates of asthma, type 2 diabetes, inflammatory bowel disease or IBD, and milk allergies. Now, there are studies that are done on mouse models, and one of them is that where they show that antibiotics reduced key bacteria like lactobacillus and segmented filamentous bacteria, and blunted one of the most important immune response, Th17 in the gut, and increased food allergy sensitization. Now, breast milk would also shape how the infant microbiome would develop. Usually, breastfed infants are enriched in bifidobacterium and lactobacillus, which produce short chain fatty acids and lower the gut pH. And this also works really well because infant specific bifidobacterium can actually digest human milk oligosaccharides. So these are the sugars in the breast milk that the baby itself cannot break down. So these bacteria actually help digest those oligosaccharides. One last thing in this slide would be about the environment that one is exposed to, a baby is exposed to, because family members would share more similar microbiomes than with unrelated people. And studies have also found that pets also matter. Like indoor dog exposure in a year one has been linked to lower risk of preclinical type 1 diabetes through a specific gut bacterium, a possible specific gut bacterium. So we now have looked into how the developing microbial ecosystem is, but how does it actually speak to immune cells? Given the broad audiences we have today, I would very try to like wrap it up in the most simplistic manner. So microbes communicate with the host through more than their physical presence. So they transform dietary and host-derived molecules into metabolites that immune cells can sense. So one major category is the short chain fatty acids or SCFAs, like acetate or propionate or butyrate, which are usually produced when microbes ferment dietary fiber. Now, what do these metabolites do? So these metabolites can influence epithelial cells and immune cell behavior, including the regulatory pathways that help limit unnecessary inflammation. Then we have tryptophan metabolites that usually signal through what is called the aryl hydrocarbon receptor. We have secondary bile acids that can influence immune differentiation, and we also have microbial histamines that can shape inflammatory signaling. So, all in all, diet can alter microbial metabolism, and microbial metabolism can in turn affect the immune tone that we set for our body. So, when they say you are what you eat, that is very much correct. Now, there's a video I again wanted to show. This is probably the last one of the videos I wanted to show on what microbes can do for us we have seen, but the other side of the conversation, like how does the gut immune system sample what is present in the intestine without launching inflammation? Like everything that you eat, why does your immune system not launch a direct attack on that? So to see that, let's watch a quick video there. Okay. So the gut is often the body's first point of contact with microbes. So every mouth of food that you take has a lot of microbes that go on to encounter the mucosa, which is the innermost layer of the gut. Now, most microbes are usually destroyed by the harsh acidic environment that is in the stomach, but a few can make it. Now, if you see the intestinal surface, it is covered with villi, which are the function of which is absorption of nutrients. Now, it also hosts the body's largest population of immune cells. So, scattered along the intestinal mucosa are these dome like structures, which are called Peyer's patches. Now, these are enriched in lymphoid tissues, making them key sites for coordinating immune responses to pathogen, while also promoting that tolerance to harmless microbes and food that we eat. Now, the villi, it contains a network of blood vessels to transport nutrients from food to the rest of the body. And embedded, we are seeing the villi right now. And then the other thing I wanted to mention is the lymphatics. From both the Peyer's patches and the villi, they drain into the mesenteric lymph node. And the epithelium, with its thick overlying mucus, that is what forms the barrier against microbial invasion. Now, one more thing, if you see while the video progresses, is that embedded in the matrix of the Peyer's patch is a mix of immune cells, which are here like B cells, T cells, macrophages. And what I want you to emphasize on here is that in the matrix of the Peyer's patch, a key function of it is, or a key function of the Peyer's patch is the sampling of antigens. So it samples antigens and then, in this case, bacteria and food bits. And to facilitate this, the Peyer's patch has this specialized phagocytic cell. This is called an M cell, which transports materials across the epithelial barrier via what is called transcytosis. Now, finally, the dendritic cells are able to extend their dendrites between the epithelial cells to sample these antigens that are then broken down and used for presenting to lymphocytes. Now, sampling antigens in this way typically results in what is called tolerogenic activation, where the immune system initiates an anti-inflammatory response. So what just happened here is that with their cargo of antigen, these dendritic cells will now traffic to the T cell zones of the Peyer's patch. Now, upon encounter with specific T cells, the dendritic cells convert them into an immunomodulatory cell which I was talking about, which are called the regulatory T cells. These regulatory T cells then migrate to the lamina propria of the villi via lymphatics. Now they have entered, and here the Tregs secrete a molecule which is called IL-10 or interleukin 10, which exerts a suppressive action on immune cells within the lamina propria. So IL-10, what it does is that it prevents any unnecessary inflammation and leads to tolerance to the food that we eat and all the good microbes that we have. Now, what happens if there is a breakdown in this process? So see how the membrane is breaking down. So what happens in this case is that the immune homeostasis tips off. There is an uncontrolled inflammation that happens there. And this is one of the things that we observe during inflammatory bowel disease, and where everything goes for a toss. And you can see how what we were seeing as a perfectly balanced, tolerant environment has now turned into something which is more inflammatory. So this is very important because it's important to understand how our gut tolerance good microbes and it reacts to something which is not good. Okay, going back to the slide.
Can you repeat that once again? How our immune system tolerate a good microbe and differentiate from the pathogenic?
Sorry, Dr. Prakash, your voice was cracking.
Can you repeat that? How our immune system tolerate a good microbe from the pathogenic or bad one?
Of course, yeah.
Now,
So
is it clear?
yeah. So usually what happens, if I say everything in short, is going to be that there are specialized dendritic cells which would present these good microbes or the food that we eat in this like tolerance promoting environment, and that favors differentiation of the Tregs or the regulatory T cells, and these Tregs would then suppress anything which is excessive effector response via like the production of IL-10 or TGF beta, and that limits inflammation. And because our gut is constantly exposed to these good microbes or the food we eat, our immune system builds tolerance over time to these different food or like the microbes that we have in our regular system. So that is what I would like say in brief, but I'm happy to like talk about it in details if anyone wants to know a little bit more. Does that answer your question, Dr. Prakash?
Yeah. To some extent, it is quite okay. Just actually, I was wondering because we take a limited useful bacteria in
Right.
early stage of life. First is of course bifidobacteria and lactobacillus, right?
Right.
But then when we use them as a probiotic, then we consume actually a large number of different lactic acid bacteria which are bifidobacteria.
Right.
So our immune system recognizes that they are also belong to the same class.
Right. Yes, it's
I hope you are
it's
clear
how
what I said.
yes. I did not catch
And
the last part.
perhaps we see the adverse effect also are what we call it the immunocompromised people. So their even
Yes.
probiotics actually induces the immune response. Yeah,
Right.
inflammatory marker. So I think that may be the
Yes.
one. Yeah, please
Yes.
go ahead. Please go ahead.
No. I think thanks for adding to that discussion. And definitely, like probiotics help. They would like introduce like, and I would talk about a few other examples of how, in like very severe cases, what is done, which is not like the very general way of treating, but it is done in like many other countries, which is the fecal microbiota transplantation. So we'll talk about that as well. But thank you for adding the probiotic aspect to it.
Yeah, because it so happened last year when I was in US and I took some probiotic samples, and it was rather than reducing the inflammation, we were actually inducing the inflammation in mice, and and it was difficult for me to explain to that professor what may be the
Yeah.
reason. So I think what you are telling is
Actually, I will
something like
also
that,
like to
perhaps.
supplement
Or like
one
iron,
point.
please.
I'll also like to supplement Dr. Prakash that it is
Please.
probiotics along with that prebiotics, symbiotics, and nowadays
Yes.
they are putting a lot of emphasis on postbiotics also. So
Exactly.
all these are very helpful.
Yes, yes. And yeah, I think like very important points mentioned. And for like students who are like new to these terms that Dr. Renu and Dr.
Exactly.
Prakash
Yeah.
were mentioning. So I would like very shortly like mention like prebiotics are these like compounds which are used by beneficial microbes. So they can be like fermentable fibers or anything. And the probiotics that we know, these are like the live microbial strains that have demonstrated health benefits. So a combination of a prebiotic with a probiotic is actually helpful, like Dr. Renu was
And also
mentioning.
postbiotics.
Postbiotic
There
as
is
well.
a different metabolites of the probiotics. And
Exactly.
also recently another new technology has come, which is called as fecal transformation. So from the feces, what they are doing is they are transforming the beneficial bacteria and putting it for the therapies.
Hmm. Yes, I actually have a slide on that later on. So thank you for mentioning that. I can show it to everyone.
Okay.
Thank you, Dr. Renu and Dr. Prakash
Yeah, please
for
go
sharing.
ahead. Thank you.
Yeah. So yeah, now since we're talking about the gut and what it can tolerate, let's talk a little bit of about antibiotics. And we all in this room would appreciate how we are talking more about antibiotic resistance these days. So the question here is, many of us have felt off after an antibiotic course, even once the infection has resolved. Why might that be? So we know that one of the possibility and a very important reason is that the antibiotics are not just killing the pathogen or the bad bacteria, but what they're doing is they're also killing the good bacteria in your gut. So this is not an argument against antibiotics. It is a statement to ensure that we use them appropriately only when they're needed at the correct dose and duration. And we should also recognize that they can affect the whole microbial ecosystem and just not the pathogen you want to target. Now, coming back to a disease in the gut that we saw in the video at the end, which is inflammatory bowel disease. This is from one of the papers which I've cited here, Wang et al. 2024. So, inflammatory bowel disease is one of the clearest examples of a disrupted host microbe relationship, but it is also one of the most complex. And what they have observed in IBD is that there is an altered microbial diversity that shifts these particular groups of organisms. So, what happens is there's a context dependent ecological change. There is also host genetics that can matter because in IBD, the NOT2 gene, it is the strongest and the most well-known genetic risk factor for Crohn's disease, which is a major type of inflammatory bowel disease. Now, if you see here, what is happening is that CD is Crohn's disease, UC is ulcerative colitis, and CTL are healthy participants. You see how many proteobacteria, it's difficult to say the numbers because 16S RNA sequencing cannot give us absolute numbers as such. But if you see the relative abundance, you'll see how the amount of proteobacteria is probably higher in CD and UC in comparison to healthy participants, whereas healthy participants have a higher abundance of like firmicutes, and they sometimes also have higher abundance of lactobacillus strains. So this is just a snap of what it can look like when we do like 16S RNA sequencing to find out what is the bacterial groups that are present in different conditions. So it would like kind of highlight the substantial alterations that we see in the diversity of the gut microbiota. Now, another disease I wanted to mention where we can appreciate the role of diet and microbiome is probably cardiovascular diseases. So, a substance which is called trimethylamine N oxide or TMAO, it is produced when your body digests red meat, and that can raise the risk of cardiovascular problems. So, the question is, can what we eat change our microbes enough to affect our heart? Maybe. And this is where I wanted to show this diagram. So dietary nutrients, including like choline or carnitine, they can be metabolized by certain meat-eating gut microbes. So there's a microbe which is called Emergentia timonensis that can convert this dietary nutrients to trimethylamine or TMA. Now the liver converts TMA into TMAO that I was talking about, the trimethylamine N oxide. And this trimethylamine N oxide has been associated with atherosclerotic cardiovascular risks. However, I want us to know that this is not a very simple story where eating one food would produce a disease, because individual microbial composition, the overall dietary pattern, host metabolism, and other factors which would also influence this pathway. Now, the broader point is that some dietary exposures become biologically different after they pass through this microbial metabolism process. Now, this is what Dr. Renu was mentioning, the fecal microbiota transplantation. So, the idea here is that if disrupted microbial communities contribute to diseases, can we therapeutically alter a microbial ecosystem? This is not a very approved treatment at all for all conditions. It is usually one of the last, I would say, like clinical interventions that come into play in conditions such as Clostridium or a C. difficile infection. So, what is a fecal microbiota transplantation? So, what happens is that it transfers carefully screened donor derived microbial material or feces into a recipient. So, you'll see how the stool is collected from a healthy donor and then it is transplanted into the intestine of a patient with the infection. And then the organism from the donor sample, they usually come in and then they would colonize that area, hoping that they would be the good microbes, and then they would restore a healthy gut microbiome in the patient. So it is a clearly established clinical role in recurrent Clostridial C. difficile infection. But like I said, it is not a generic microbiome reset. It involves donor screening, manufacturing, route of administration, patient selection, and definitely safely monitoring. So, how it is done, it can be done in different ways. One is how it can be inserted by a colonoscopy, and then there are also different other ways that have come up where pills have come up where someone can take a pill which would promote a fecal microbiota transplantation. There are two other different ways as well in which it is done, but inserting it via colonoscopy is probably one of the most common ones. So, a bit of a big picture thing that I would want everyone to leave with is that first, microbes are not simply our enemies. So, most host associated microbes are either harmless or they benefit in the usual context and help our immune system run constantly from the moment we are born till the end. Second, the immune system's goal is to balance. So, it must protect us from pathogens while allowing peaceful coexistence with the microbial communities that we really depend on. And third is when the conversation changes through development or diet or infection, antibiotics, or any barrier damage, the consequences actually can extend far beyond the gut. So, the microbiome is a dynamic ecosystem that helps shape how the immune system learns to tolerate everyday items like food, good microbes. It helps to repair and it helps to defend. So, with that, I hope there was a brief introduction as well as going into different disease models that we understood. Thank you very much again to everyone who has been here listening to me for the past 45 minutes. And I would again like to thank everyone and everyone who supported me throughout my journey as well. I'm happy to connect with everyone after this talk and take any questions that you have, and very open to having a discussion. And if anyone wants to add, like Dr. Renu and Dr. Prakash were adding, please feel free to take the stage and add those. Thank you.
Hello, ma'am.
Hello. Hi, Vedanti.
Ma'am, I wanted to know about something about your research topic. Like you
Okay.
mentioned that it is about acute inflammation after spinal cord injury. So, can
Mhm.
you just tell what is exactly?
Yeah, sure. So, what we look at in the lab is basically so after spinal cord injury, we have what is the primary injury phase, which is when there is that initial mechanic disruption to the spinal cord. which is followed by what is called a secondary injury. So during this secondary injury phase, we see a lot of edema, like where there is swelling. We see a lot of nerve cells that get damaged, a lot of, but the most important thing that I want to emphasize is that we see infiltration of a lot of immune cells. Now, when there's a lot of immune cells that get to the site of injury, that can prolong the recovery phase. So one of the things that I was looking at was a molecule which is called L-selectin. So L-selectin is this molecule that usually helps one of the immune cells called neutrophils to roll on the surface of the vascular endothelium and get to the site of injury. Now, usually when these immune cells get to the site of injury, they would usually get activated and the L-selectin is shed off. But my question was, one of the things I'm looking at currently, this is not like my final PhD project, but this was one of the aspects I've looked at and presented on, is what happens if L-selectin cannot be shed off by these microbes, by these immune cells. What happens then? So what we found was that if L-selectin cannot be shed, we used what is called an LE mice. So LE mice have a mutation that does not let L-selectin shed off from the surface of immune cells. What we saw was if LE mice or mice that cannot shed their L-selectin, they had a lot of more immune cells such as cytotoxic T cells. They had more number of neutrophils at the lesion epicenter. So what we are looking at is how are different immune cells after the injury, whether it's at acute time points or if it's at chronic time points like 35 days. I work with mouse models. So I wanted to see how different immune cells either benefit, either help in recovery, or are they causing more damage and slowing down recovery. We test a lot of drugs. We would also do a single cell RNA sequencing. We would do a lot of flow cytometry to find out what is happening to the different immune cell populations in different conditions. The major thing that I look at is also sex differences. How does it differ between males and females? So yeah, that is kind of a brief on what we do. The techniques I use is also I do a T9 contusion surgery on the spinal cord, which involves doing laminectomy, which is I think one of the most difficult things I've found, or one of the most difficult techniques I've done so far in life, honestly, because when you do a laminectomy where you remove that lamina from over the spinal cord, you have to be very careful because otherwise you might nick the cord. So we do a laminectomy, and mice are really small, right? So doing the whole surgery under the microscope. So after that, we give a T9 contusion of a particular force severity. I do 60 kilodynes. And then the contusion happens, and then the mice lose function from the hips. So they also need bladder care twice a day. We do the best that we can do for those mice, and then we observe their behavior for 35 days. And then we do other different functional assays as well as part of it, which also involves a lot of immunostaining that I've done so far. I don't know. I just went a little too much into the details, but I hope that answered your question, Vedanti.
Yes, ma'am. Thank you.
Of
I
course.
asked you because I thought that your topic is very new and interesting.
Oh, thank you. Thank you. Thank you.
Yes.
Appreciate
Thank
it.
you, ma'am.
Of course. Anyone else who has any questions, or if anyone wants to talk about anything, discuss anything, anything new that you have found in the field that you want to share with others?
Hello, ma'am. Am I audible?
Yes, Gayatri, you're audible.
Yeah. First of all, ma'am, thank you for this insightful session.
Of
And
course. I'm happy.
yeah, moving to my question, ma'am. I completed my MSc in general microbiology, and
Mm-hmm.
I studied subjects such as agricultural microbiology, food microbiology,
Mm-hmm.
bioinstrumentation, and many more. So, however, I don't have many research publications yet.
Uh-huh.
If I want to pursue a PhD abroad, especially
Mm-hmm.
in Canada, what should I
Mm-hmm.
focus on to strengthen my profile? And should I take any additional courses, gain research experience, or improve specific technical skills? Or are there any other things you would recommend before applying?
Yeah, so do you have any poster presentations that you have done, Gayatri?
I attended, ma'am. Like
Oh,
I didn't participate.
Okay. I would suggest participating because I think PhD programs abroad, it gets even more difficult with each passing year given the funding scenarios and everything. So it becomes important to have a very strong research background. So one thing would be definitely if you can come up with a research project, that would be great. If you could work with one of your professors and like do a short research project that you use different techniques for, that would be ideal. The
Okay.
other thing you can do, yeah, so you can also write a review paper with your professors or some of your batchmates. That is
Mhm.
something you can do to have publication up your sleeves. If not, that is fine. A lot of people who apply for PhD programs do not have a publication, and that is okay, as long as you have enough research experiences. Now, another way of boosting your application is definitely attending, not just attending conferences, but presenting in conferences, because that shows them that you can communicate.
Yeah.
Yeah, you can communicate your science. That's how I would put it.
Okay. Okay, ma'am. The communication skills must be like good now.
Yes, and then having research experience would definitely help. So try getting research experiences however much you can, because that would highly strengthen your application. Because one of the things that you have to put in statement of purpose is going to be the usually the second paragraph. There's no like norm as such that it has to be in the second paragraph, but majority of your statement of purpose would have to talk about the research you have conducted and how that links with the school you're applying to, or like how does that influence your research decisions, or like how does that influence what you're going to study or what you're trying to achieve with a PhD. So research experiences would be very beneficial. Honestly, if needed, if we get a lot of these questions from students, I do usually on LinkedIn a lot of posts and a lot of seminars on how students can apply for masters, even like for postdoc positions and everything, PhD positions. So I can with MBSI, I can talk to the committee if they want. I'm happy to put up a session on discussing these options and what you can do, what you
Yeah.
need for a CV or SOP or things like that.
That would be really grateful, ma'am.
Okay. Thank
And
you,
it's
guys.
really helpful. Thank you. Thank you, ma'am.
Of course, I
Ma'am,
need.
yeah, ma'am, hello.
Yes, I can
I'm
hear
having
you.
one, yeah, ma'am. Thank you very much for your very much informative session. Actually, I will miss it out and later on join it. But anyway,
Mm-hmm.
I'm from pharmaceutical background, and
Okay.
I'm interested to have kind of formulation where the
Mhm.
probiotic can play a vital role for enhancement of the viability of calcium. So, do
Okay.
you have any kind of professor or researcher, those who are working on probiotics, like any
Mm-hmm.
of these strain of probiotics, which can
Mm-hmm.
enhance this calcium salt viability? So
Mm-hmm.
I'm looking forward for your help if somebody can connect me or you could help out so we can do something better for the society because you know that most
Mm-hmm.
of the time the calcium salt viability is hardly achievable like up to 40-50%. Rest of the
Right.
calcium is just like a wastage for our body. But somewhere I've read it out through literature, it's showing some great influence on the absorption capacity of the calcium salt with
Yes, yes.
mild incorporation with the probiotics.
Right, right. I know that there is a symbiotic effect as well, and then it can
Yeah.
also like yeah reduce bone resorption. So that is
Yes.
great.
Yeah,
So
ma'am. Uh,
I I can think, but if there's anyone in the audience today, if you would want to connect with Gati, that would be nice as well. And let
yeah.
me get back to you. Let me see if I have anyone who's working
Yeah.
in
Yeah.
this.
Yeah, but related to calcium bioavailability, just yesterday only I made a one project presentation using PGA, polygalactouronic acid, and making
Okay.
encapsulation with that, which actually helps for the slow release and
Okay.
targeted delivery. So
Means
you
by
can actually use
encapsulation. Yeah.
exactly
By
with PGA.
encapsulation.
Yes,
Okay.
yes. And otherwise also we use the probiotics for the several of this mineral bioavailability, especially through the phytic acid degradation process. You know,
Okay, sir.
Because what happens
Mhm.
is that most of these minerals and trace elements, no, they will be bound with the phytic acid and
Okay.
will be in unavailable form. So we
Yeah.
have actually this project on this aspect where we look the bioavailability. We have actually several of these publications, and we are working on this anemia now,
Oh, sir.
through this similar approach.
Oh, sir. Okay.
Exactly.
Yeah.
So
Good to
anemia,
hear
of
you,
course,
sir. Mhm.
anemia project just 1 month back we submitted to ICMR,
Okay,
and if
sir.
that result comes favorable, we are somewhere some 4 or 5 institutes, including NIN, National
Okay.
Institute of Nutrition, who will be
Okay.
participating in this project, where
Oh,
we actually make some 2-3 different combinations
okay.
and look for the bioavailability of the several of the stress elements as well as this vitamin,
Okay, sir.
because
Yeah.
folic acid and vitamin cobalamin are actually responsible, no, for this anemia also.
Right,
So,
sir. Right.
exactly.
Yeah. I will definitely get connected to you, sir.
Yes,
Definitely.
definitely. Yes.
Your good name, sir? I'm not looking, sir, in this. Your good name, sir?
Prakash Halami from CFTRI Mysore.
Okay, sir. Okay. Thank you so much. Yeah, sir.
Thank you. Thank you.
Thank you. Thank you. And thank you, ma'am. Yeah.
Of course, yeah. I'm glad you found the connection. Wanted.
Yes. Avantika,
Yes,
there are
Dr.
any questions from the students? Just I had a general comment actually to make.
Mm-hmm.
Can you hear me?
I can hear you. I can hear you.
Oh,
Yes.
wonderful. Yeah, actually we call probiotics, actually they train our immune system,
Mm-hmm.
right?
Yeah.
So what do you mean by how you explain in the students from the student perspective?
Yeah, so I think if I have to talk about how probiotics can train immune
Train immune
system,
system. Yes.
yes. I would say there wouldn't be a guaranteed pathogen specific immune memory, but these probiotics and the molecular products that they bring with them, they would definitely interact with epithelial cells and the gut associated immune cells that we saw in the video. And
Yes.
the pattern recognition receptors, like the toll-like receptors, they would detect these signals and shape the type of responses that are formed. So, like for example, some of the probiotic strains we know that can actually stimulate mucus or like the tight junction integrity, and that can
Mm-hmm.
reduce pathogen access to deeper tissues. Or then there can be like some Treg associated responses that they favor. So that's how I would put it. These are some of the mechanisms that they can bring into the picture.
Exactly. The other question is, or I would
Yeah.
say comment. See, actually,
Mm-hmm.
probiotics regulatory agencies, they don't recommend for children below 5. But
Mm-hmm.
if we see their immune system actually gets again
Mm-hmm.
matured because of this probiotic bacteria, right?
Mm-hmm.
Probiotic bacteria or gut microflora.
Mm-hmm.
So what is your opinion? That the infant should take a probiotics or the healthy microflora in their GI tract, or they should not? Or it should be a natural process, you mean?
Um,
No, like natural means whatever it comes through the mother's milk,
mm-hmm.
whatever it comes with the infant formula, that one. But if you see, we had actually a good study, especially from Orissa by Panigrahi. They
Mm-hmm.
actually did for prevention of the sepsis, you know, in a small
Mm-hmm.
children. They
Mm-hmm.
did this work and they could publish in Science, I believe, that paper,
Mm-hmm.
2009 or 2010.
Mm-hmm.
Most of the authors are from actually US, but that
Mm-hmm.
study was actually done in Orissa
Mm-hmm.
among the children who actually used to have the infant mortality, and then they could actually save several of these children through this approach.
Right. I think, yeah, I mean, I know that it's very debated, like whether it should be given. I mean, I know that there's no harm as such, but I think
Mm-hmm.
it comes more from the point that people think like the natural microbiota that, or like, how in human milk there's serum immunoglobulin A, there is a lot of like beneficial factors that go in. So, and then the immune system is also pretty naive in very young kids. So
Yeah,
how
exactly.
yeah, so I think it's based off like a balance where they think that if there is um any microbe that can probably like colonize in these like probiotics, which I don't think it is the case, because probiotics in general would never have, or would ideally never have, anything that would disrupt the immune system or like disrupt the microbiota of a growing young baby. But like you said, I think, yeah, it's a trade-off that they usually do when
Yes,
it comes
yes,
to
yes, yes, yes. Yeah,
scenarios.
just like I think, like immunocompromised patients or the
Exactly, exactly,
even for the pregnant woman also we don't prescribe the probiotics.
exactly. Yeah, I think it's more because of
Unless
that.
and otherwise the medical doctors actually prescribe. Under the only can be under medical doctors this supervision. Otherwise
Yeah,
we don't. Mm.
yeah.
Very
I
good.
think it. Yeah. Thank you for sharing that.
Thanks. Yeah. Thanks.
Okay. Anyone else who has any other questions or comments? Yeah, if you have any questions after the session, I'm very happy to connect. We can connect on LinkedIn and we can talk and discuss about anything related to microbiome or the immune system. Happy to answer those questions.
Yeah, just I have one small comment. Actually, in fact, it so happened last year
Uh-huh.
when I was in US, I visited Cleveland Clinic, and
Mm-hmm.
they did this TMO related work.
Right. Okay.
Especially
Mm-hmm.
the TMO metabolism, and then that professor who was doing, he said that
Mm-hmm.
time there was no any studies from the gut microbiota perspective.
Mm-hmm.
Okay,
Yeah.
the gut microbiota was investigated especially on atherosclerosis or this cardiovascular diseases afterwards. But
Right.
then they actually could publish that time in Science, and then could make actually the link with this what we call the red meat, right?
Right. Right. Yeah.
The consumption of the red meat, how actually it triggers the atherosclerosis and all these things. So that was
Mm-hmm.
actually very useful finding. I believe that professor name is Hadley, I guess. He is actually the
Okay.
director of
Mm-hmm.
Cardiovascular Research Institute in Cleveland Clinic. And
Mm-hmm.
also now most of these such diseases, they are actually linking with the gut microbiota, and one of the organism which you were explaining, I think that was also important, I believe.
Mm-hmm.
Yeah, because this
Yeah.
kind of organism now they are using as a next generation probiotics to
Right.
target a
Right.
specific diseases. Yes, just a small comment from my side. Otherwise,
Thank you.
thank you very much, and good luck for your PhD.
Thank you so much. Thank you so much.
Yeah,
I
yeah.
appreciate everyone and the discussions we've had today.
Thank
Thank you,
you.
everyone. Yes. And if we have no more questions, I'll take a leave. But yeah, hoping to interact with all of you very soon again. Thank you. Bye bye.
Anyone is there to conclude the session, or it is just a getting
Yes,
off
sir.
automatically? Yeah,
Yes, sir.
please. Yeah, please
On
go ahead.
on behalf of NRI lecture series, we are
Wonderful.
thankful. We are thankful to our president, Dr. A.M. Deshmukh sir. We are also thankful to the resource person, uh, Abantika Ghosh ma'am. We are also thankful to scholars, professors, and the dignitaries who joined this lecture and listen carefully and make this session very interactive. And last but not the least, we wish the resource person Abantika Ghosh for his PhD. We
For her.
our best wishes with her and
Yeah, exactly.
for her future endeavors.
Yeah, exactly.
Thank you very much, and have a good day.
Thank you
Thank
so
you.
much.
Bye.
Thank you so
Yes.
much. Bye bye.